This is the McMush Macromolecule Food Lab. The question which we are trying to answer is which macromolecules are present in the McMush food slurry? Our hypothesis is that if carbohydrates, proteins, and lipids are present in the food slurry, then each of these will test positive in the presence of their respective reagents. We will start by doing the lipid test. For the lipid test, we're going to put approximately 1 milliliter of vegetable oil into one of these wells on the well plate. I'm actually going to put this well plate over a piece of paper towel so that way we can see the reactions take place easier. So in the first well plate, I'm going to put about 1 milliliter of vegetable oil. The next one will get approximately 1 milliliter of distilled water. Oops. And the third will get approximately 1 milliliter of the McMush solution. I will then add in a few drops of the Sudan 3 solution, so approximately 5 drops into each. So 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. If you look carefully, see if that light helps at all. You can see some little clumps in the well that has the vegetable oil in it. So you can see some of that orange clumping in there. In the middle one, that's with the distilled water, there is no clumping. If we look carefully at the McMush solution, you can see a little bit of orange clumps forming in the lipid test. Because we see orange clumps forming, that means that this is a positive result. So we have our known vegetable oil, we know that there's lipids, and we see a little clumping. We know that water does not contain lipids, so there's no clumping. And in our McMush test, we can see that there are some little clumps, and that means that lipids are present. For the protein test, we will test some egg solution for our known positive. So put about 1 milliliter of egg solution into the well plate. Then again, we have our known negative of distilled water. And in the third one, we will put our McMush. To test our egg solution, or to test for proteins, we are going to use Biurette's solution. So we'll put a few drops of Biurette solution into each. So we'll put about 10 drops. And we'll make some careful observations here. Let's zoom in. In the egg white solution, we can see that it turned a purple color. In the water, it stayed blue. In the McMush solution, we're not seeing a big color change happen. If I give this a little bit of a stir, I'd say that's not purple. It looks a little bit greenish, but I would not say that this is a positive result for the protein. So remember, a positive is the egg white, where it turned purple. A negative is the distilled water, where it turned blue. And I'd say that this McMush solution is inconclusive. We cannot definitively say yes or no to the protein in that particular scenario. The next test is for polysaccharides. To test polysaccharides, we will use a starch solution. So I'll put some starch solution in the well on the right. So we've got starch solution, about 1 milliliter. Then we have distilled water in the next one. And McMush finally in the last. To test for the presence of polysaccharides, we will use Lugol's iodine. So you need about 3 drops of Lugol's iodine in each. So got 1, 2, 3. 1, 2, 3. 1, 2, 3. In the known positive test, so in the starch solution on the bottom, it turned a black color. In the known negative, so in the water, it turned an amber color. And in the McMush, it also turned a dark black color. This tells us that the McMush solution must contain polysaccharides. For the last test, we will test for monosaccharides. In each test tube, we'll use 3 of them. We will have our known positive, our known negative, and the McMush. Our known positive is a glucose solution. We'll put approximately 3 milliliters of glucose into the first test tube. 1, 2, 3. So approximately 3 milliliters of glucose solution into the first test tube. We'll then add 5 drops of the Benedict solution to that test tube, and then place that test tube into the hot water bath. So 1, 2, 3, 4, 5. The second test tube gets approximately 3 milliliters of distilled water. It also gets 5 drops of the Benedict solution. The final test tube will get McMush added to it. So again, approximately 3 milliliters of McMush. So we've got the McMush in the third one, and that will also get 5 drops of the Benedict solution. Right, so we have our starch solution going into the hot water bath. Then we have the water solution, and finally the McMush solution. We will let that sit in the hot water bath until a reaction takes place. The test tube on the far left is the glucose solution. The test tube in the middle has distilled water, and the test tube on the far right has the McMush solution. as you can see, as time goes on, the glucose solution has turned a reddish orange color. The distilled water solution has stayed mostly clear with a slight blue tinge, and the McMush solution is starting to turn a orangish red color.